Hello, ladies and gentlemen. Our project is development of a model for prediction-based sequence mutation in COVID-19 using transformer technique. The COVID-19 situation in 2019-2020 has 285 million cases and 5 million death cases, causing the global economy to collapse. But eventually, economy came back for better. When the number of infected people declined, COVID-19 is spreading again, resulting many in more infection and many more deaths in a little time. What is the reason for the increase in the number of death and infection? The cause of this problem come from is a small point. It is called a mutation. Mutation to occur in nucleotide sequence. There are two mutations in the COVID-19 virus. Case 1 mutate and didn't survive in the normal environment, in which case is not important. Case 2 is survival in the environment. This will make the property of the virus is better, such as being able to infect more easily, and may result in more difficulty to controlling the spreading situation. Therefore, we have an idea to develop a model for predict the mutation of COVID-19 nucleotide sequence using transformer technique. For the researcher can take it better by looking at the structure and property that may occur in the future, and make plan to control the situation in the future is better.
This will be an overview of the operation. We start by collecting data with a FATA file from KitH and NCBI. Then do a data processing and encode the data so the computer and can predict it. In encoding, we will study how to tokenize the computer able to understand the most information. In the second lab, we study DNA with different external factors. Then take the model let's learn with the lasted data range and predict new sequence. In pre-processing, we will do alignment to sort the index so that the range of data matches every sequence, but the alignment will have some range of sequence with that and n, which does not affect the mutation, so we cut it out and we arrange the list. We take every sequence in that data range and bring n-camera tokenization in each token, then slide one by one until the whole dataset is complete. In experiment 1, the bird pretend was fine-tuned using different tokenized methods. We measured with perplexity and F1 score. The best model was used in the second experiment. In the second experiment, bring three copies of information within the affected period. Then, let the model train the data in the original architecture to measure the complexity of the data with infected data. Thus, model performance which represents the mutation. To create a new sequence, we took the best method, fine-tuning with the lasted data and predicting a new sequence. We take all sequences of DNA to find the same codon of the same index and leave the difference for the model to predict. So we get a new sequence with an empty point, then do a camel tokenization of the whole sequence for the model to predict and compare with the answer, so the result is as follows. Accuracy uh, by was 91.7%. Accuracy by character 97.7%. One score 83.9%.
Next, this will be the summary of the experimental result. In the first experiment, studying the efficiency of different tokenization with market language modeling using a fine-tuning technique. From the result of the experiment, we found the DNA-based with 6 tokenization has the highest perplexity and highest F1 score. Finally, the DNA-based model is the most efficiency. That is because it has more tokenization. As a result, the model learns the dataset better because of there are many patterns to study, causing the model to confuse the dataset with less and achieve the highest efficiency. Next, in the experiment 2, we found that using the data in December, which had the lowest number of infection, result in the model learning is the best. But in February and January month, they have the higher number of infection. As a result, model learn less than December because the more people infection, the virus can change more hosts, resulting in virus have many mutations. It's not surprising the model is effective rate because of the more complex data are many.